assistant professor in the Department of Biomedical Science, Creighton University. And also I'm the co-chair of this Bellucci Symposium. And this year, we focus on inner-ear development and the sensory hair cell regeneration. As many of you know, the lack of regeneration capacity in the mammalian cochlear leaves millions of people suffering from irreversible hearing loss. We are hoping to find a therapeutic treatments to regenerate sensory hair cells to restore the hearing function. However, there are so many questions need us to answer before we can find such a regenerative treatments, which require us to look into the process of inner ear developments. So we want to know what signals provides the temporal and spatial informations for the cochlear to develop, and we want to know what transcription factors dictates the hair cell differentiation process and specify the hair cell subtypes. And what we can learn from the regeneration process in other inner ear organs, such as utricles, and what does it tell us that non-mammalian animals like birds or fish, they can regenerate sensory hair cells or to restore the auditory functions. And whether we can take similar approach people take to regenerate retina, to regenerate heart, and we know cochlear is small, it's difficult to dissect. Whether we can find a better model to study the hair cell development and regeneration. With these questions in mind, we invited a group of great scientists today to share their exciting work in inner ear development and hair cell regeneration, as well as retina regeneration and heart regeneration. There's no doubt talks today will stimulate more questions, inspire new ideas. So first, let's give a round of applause to all the speakers. Now I will give the stage to Dr. Junxia, the moderator of morning session. Enjoy the talks.
Good morning, everyone. I'm Junxia, Assistant Professor of Biomedical Science, and it's my great pleasure to be the moderator for the morning session of the Bellucci Symposium. Today, we have a great lineup of speakers, as Litao mentioned, and the topics of the talks that we'll have this morning include single-cell RNA-seq and using multi-omics approach to understand cochlear and inner ear development. And also, we have talks covering the transcription factor roles in vestibular hair cell and signaling in cochlear and spiral ganglion cell development. It's my great pleasure to introduce today's first speaker, Dr. Matthew Kelley from NIDCD. Dr. Kelley received his bachelor from Cornell University and PhD from the University of Virginia. And following postdoc fellowship at the University of Washington, he became an assistant professor in the Department of Cell Biology at Georgetown in 1996. And in 1997, he received the KO2 award from NIDCD. And 3 years later, he moved to NIDCD and first as acting chief, and then in 2004 as the chief of the developmental neuroscience section. In 2010, he became chief of the laboratory of cochlear development. Dr. Kelley's laboratory works on the cellular and molecular mechanism of the mammalian cochlear development. And he has received numerous awards, including the 2016 NIH Director's Award in honor of exceptional performance and special efforts beyond regular job assignments in NIH. And he was past president of the ARO. He's currently the ARO program chair, and he serves on many committees, including the NIDCD rank and tenure committee. And today he will talk about the identification and transcription regulators of cochlear development using single-cell RNA-seq approach. Let's welcome Dr. Kelley.
So good morning. Thank you for the kind introduction. And I want to thank the organizers for giving me an opportunity to kick off what's going to be a fantastic symposium once we get through this first talk. So as most of you know, my lab is interested, you just heard, in development of the organ of Corti. And we have a longstanding interest in trying to understand how cellular differentiation and specification occurs within the epithelium. So what I'd like to do today is just to give you a very brief introduction on organ of Corti. I don't think we need to spend much time on that. Talk a little bit about single-cell RNA sequencing as a new way to really move the field forward. Go over a study we did just a few years ago, trying to take a first step towards using single-cell RNA-seq to identify new signaling factors within the organ of Corti, and then show you a couple of genes that we've identified through that we're now sort of working out on figuring out what they're actually going to do. Okay, there we go. Okay, so up here on the top is a paint fill from Doris Wu. I've taken a little bit of liberty. It's not really a P14. It's probably considered a little bit younger than that, but it very nicely shows the structure of the inner ear. And here's the cochlear spiral. If we were to cut a cross section here, we see this lovely structure that we're all very familiar with, the organ of Corti, which contains two types of hair cells, inners and outers, shown here in red. And then a group of associated cells referred to as supporting cells, which is a fairly amorphous term and basically stands for any cell that's close to a hair cell but isn't a hair cell. There's some that we can very clearly name based on their morphology: interphalangeal cells and border cells here, inner and outer pillar cells, Dieter cells, and Henson cells. But then there are other cell types here, Claudius and Butcher cells, and Colliker's organ or inner sulcus cells here that some people consider supporting cells and some people don't. But for the purposes of this talk, we'll stick to these cell types here, which have very unique morphologies. Now, if we were to look at the same region of the cochlear duct at an earlier age, in this case, E14 in a mouse, again, another paint fill from Doris, you can see the cochlear duct is just beginning to grow out at this stage. It's about half a turn. And if we were to take a cross section, we see a very different looking epithelium. So the epithelium in this case is essentially a classic pseudostratified epithelium. And if we use genetic sparse labeling, we can see that the cells, in fact, have very clear epithelial undifferentiated cell types, extending both luminal surface and basal membrane processes with a centrally located cell body. The cells here located in the yellow box are referred to as prosensory cells. And these are the cells that we believe will go on to give rise to all the cell types within the organ of Corti. Now, we can't say for sure, but the general feeling in the field is that at this stage, these cells are essentially equipotential in that they have the ability to give rise to any of the cell types within the organ of Corti, either hair cells or supporting cells. And so what that means is that between this time point and this one, these cells have to make a decision about what type of cell they're going to become, and then activate a transcriptional program that will drive their unique physiology and morphology. And that's illustrated here. So we have these equipotential prosensory cells. They have multiple choices that they can make in terms of the cell type that they're going to become. And their subsequent cell fate commitment and differentiation must be driven by genes, and in particular transcription factors. And so it's been a goal of my lab to try and understand these different signaling processes. Now, one of the problems that we've had over the years is that large format bioinformatic approaches such as RNA-seq were not well suited for an epithelium such as the organ of Corti, which has a very high number of different numbers of cells, but a very low number of actual cells. So there are only about 1,000 inner hair cells in a mouse cochlea. And so any approach in which we took the entire epithelium was not particularly well suited to try and identify genes that were specific for one cell type or another. That situation became much better about 10 years ago when single-cell RNA sequencing came on the stage. And I was really lucky at the time to have three very talented postdocs in the lab: Joe Burns, Mike Kelley, who's not related to me, and Mike Hoa. And the three of them convinced me that we needed to be out in the forefront in terms of using single cell. And I was wise to listen to them. So what I want to do is just give you a very brief overview in about 2 slides of single cell RNA sequencing, because I think most of us are probably pretty familiar with this. Basically, the first step in doing a good single cell study is to isolate the cells that you care about in terms of a good dissection and then getting a good single cell preparation from them. So what we did to start with this was we generated a triple transgenic mouse in which we could label the hair cells and supporting cells in different colors. We then did a very careful dissection to isolate just the floor of the cochlear duct. We enzymatically dissolved and removed all the underlying mesenchymal cells, which otherwise would overwhelm the single cell approach. And then we took that isolated strip of epithelia and dissociated it down to single cells. And you can see both hair cells and supporting cells and non-sensory cells here in our single cell dissociation. So once you have a good dissociation, the next step is to collect those cells. And there are a number of different ways that this can be done. The one that's become sort of the industry standard at this point is using the 10x Genomics Chromium platform. So this is a microfluidics-based approach in which we use an instrument to basically create aqueous droplets that have within them single cells and something called a gem bead. And those droplets are then created in a heavy viscous oil. So once those droplets are formed, one of the first things that happened is that the cell itself is lysed. And then the gem bead is what comes into play at this point. So a gem bead is essentially just a physical bead that's arrayed with thousands of oligonucleotides. There are a bunch of different sequences in that oligonucleotide, which are illustrated here. The two that are most important are poly T or oligo DT region here that's going to capture the poly A tails from mRNAs, and this region called a 10x barcode. Now, the barcode is just simply a 12 nucleotide sequence. But what's unique about these barcodes is that every oligo on a single bead has the same barcode, but the barcode changes from bead to bead to bead. So once those mRNAs are captured, they're converted into cDNAs, and during that process, they incorporate that 10x barcode. We then use Illumina sequencing to get back data on the 3' end of the genes that have been captured, and also the 10x barcodes. We can then align every gene that has the same barcode as coming from the same droplet and therefore from the same cell. Now, this system works fairly well for doing fairly large scale captures. We found that for embryonic and early postnatal cochlea, we load about 6,000 cells, which we can typically get from a litter of mice. We capture about 3,000 of those cells, so reasonable capture. We end up getting about 50, when we put those on the Illumina sequencer, we get about 50,000 reads per cell, which gives us data on about 2,000 genes. So we're definitely talking about a bird's eye view here in this type of approach. And because of the Illumina sequencing, we're only getting data from the 3' end. So this is really just gene counting. We can't look at isoform usage or anything like that. But it does give us a very nice overview of the cells that are present within an epithelium. So a few years ago, Mike Kelley and a postback in the lab, Lakeitha Kola, began a project to basically examine cochlear development from embryonic day 14 through early postnatal using the 10x platform. And I'll show you just some of that data here. So this is a TSNE, which is would now be considered an old school UMAP presentation of the data. Basically, every dot here, it represents data, multidimensional data from a single cell. Dots that are the same color are transcriptionally similar in nature. And we started with P1, where we generated captured about 12,000 cells. And you can see here in our P1 analysis, based on gene expression of genes we know, we were able to identify inner and outer hair cells. Interphalangeal cells are over here, inner pillar cells, Henson cells, Diter cells, and outer pillar cells. So all the cell types that we expect to be there at P1, and then a lot of cells that are here in Colliker's organ. So from this data, there are a number of things that we can do. One that I just want to take a minute to talk to you about is it's a great way to identify new markers for different cell types. And so this advances, whoops, too far. Here we go. So this is a feature plot now of the same data, and these are two genes we identified that were new hair cell markers, CCER2. So now the redder the dot is, the more it expresses the gene of interest. And here's SM fish in situ hybridization for CCER2, showing it's hair cell specific. And here's a different one, SLC6A11, also just in hair cells, and here's SM fish for that. So this has worked very well. And then what's been more useful for us actually are identifying markers for the supporting cell types. So neuropeptide Y turns out to be an inner pillar cell marker. You can see that right here. And also angiotensin 1 converting enzyme, also a pillar cell marker. You can see that here in SM fish with parvalbumin for the hair cells. And fatty acid binding protein 7 turns out to be a marker for interphalangeal cells and Henson cells. You can see that here in an immuno. So we've got FABP7 surrounding the inner hair cell here, and then in the Henson cells out here. So this data has been quite useful for identifying new markers for different cell types, which we've applied in a number of ongoing projects in the lab. Now, I want to go back to this slide here, this image here, to talk about what I really want to spend the rest of the talk on, and that is trying to identify transcriptional regulators. So one thing that was really interesting about this data when we got it was that we noticed that the outer hair cells were actually arrayed in a linear pattern here along both of the two primary axes. And we wondered what might be driving this arrangement. And one thing we thought might be the case was the fact that, as most of us know, the organ of Corti develops in a gradient that starts in the base and moves towards the apex. So at a time like P1, we're going to be collecting hair cells that are different stages of development. And we wondered if this array wasn't a reflection of that developmental process. So if we look at a couple of genes that we know are modulated during development, we can see that that's in fact the case. So ATO1, which is required to make hair cells early on, is expressed highly on this end of the array, but is going down as we move this way along this array. And calbindin-2, which is a calcium binding protein for hair cell function, it has the opposite pattern. So it's low in these cells here and higher here. And that confirms that we in fact are getting hair cells at different stages of development. So what we wanted to do was to try and build out a more comprehensive profiling of outer hair cell development. So what we did was to do the same type of capture at E14, E16, and P7. And in each of these ages, we were able to identify outer hair cells, which you can see here and here. Here's the P1 again, and here are the P7s. We can then take all the data from all of those cells, combine it into one new data set, and then essentially do a genetic transcriptional analysis where we identified the developmental gradient of outer hair cell formation based on changes in gene expression. And we did this using a program called Monocle. And so that's what this array looks like here. Now these have been, now we've replotted this a little bit. So this is our array of transcriptional or outer hair cell development from least mature to most mature. The colors here match the date that the cells were collected on, and very reassuringly, you can see the E14 and E16 cells are here. They then transition into the P1 cells here. We then have this very irritating gap, which basically told us that we should have collected P3 and P5, which we're doing now. And then here are the P7 cells that we can see here. And we can then essentially look at gene expression across this developmental process, which we refer to as pseudotime, from least mature to most mature. So because we have data on roughly 2,000 genes per cell, we can then look for patterns and changes in gene expression over development. And what we found from this was a really nice result here, where we have 4 phases of gene expression. So we have genes that are turning off early. We have 2 phases of transient gene expression, and then we have a final phase of genes that are turning on and staying on. And as far as we can tell, what we're seeing here are prosensory genes that are turning off. These 2 transitional phases seem to be, we think, the first one seems to be genes that are involved in specification of generic hair cells. So they're genes that we see turned on in almost every hair cell during development in both. I don't think I did that. Do I get more time if I have to start over? Okay, phase 2 we think is more generic genes, so genes that are expressed in all hair cells and all vertebrates. Phase 3 we think is potentially more organ or cell type specific, so auditory or outer hair cell specific. And then phase 4 seems to be more nuts and bolts types of actual functional genes. And so some examples. Here's SOX2. This is SOX2 turning off as cells are becoming hair cells. It's a prosensory gene. ATO1 turns on transiently and then goes away. And here's CALP2, which turns on as cells are becoming functional. So we can basically summarize this like this. We have 3 patterns of, we have these 4 patterns of expression we see over the course of hair cell development. Now, what we wanted to do next was to take advantage of bioinformatics to try and identify potential candidate transcription factors that might be driving these different phases. And we basically took a 3-pronged approach here. The first one was the most obvious. We just looked at the data we had and asked how many transcription factors do we actually see. One of the things that we wanted to do to complement that was one of the problems that I told you about with 10x single cell sequencing is that it really gives you a bird's eye view. And because you're only getting data on 2,000 genes, quite often genes that are expressed at fairly low levels, which is often the case for transcription factors, might not actually be expressed in the data set. But we can use bioinformatics to look at genes that are expressed within the data set and then look at the promoter regions to identify putative additional transcription factors that might be expressed. The last approach that we took was we did a separate study where we used population RNA-seq to compare cochleas from A201 wild type and A201 mutant mice. And this was going to give us insight into genes that were downregulated when hair cells are missing. So we used all three of these approaches. The obvious one I mentioned to you, David and Scenic, two of these bioinformatic analyses, and then this comparison of A201 positive and A201 negative tissue to identify a panel here of candidate transcription factors that might be expressed during these different phases of development. Now, I want to mention that this is by no means a comprehensive list. These are just the ones that look most obvious to us from this study. You'll certainly probably hear about some other ones today that are not on this list. Reassuringly, when we look at this, we do see a number of what we would consider the usual suspects. So genes that we already know are expressed. So here's SOX2, ATO1, PAL4F3, INSM1, which you may hear more about later today, IKZF2, which you may also hear more about later today. But what I'd like to do is to finish up by talking to you about two new genes that we found here that we've begun to just have a look at. So the first one is this zinc finger protein called CASI1. And this is a gene we were interested in based on its homologous role in Drosophila. So CASI1 stands for castor zinc finger 1. It's the mammalian homolog of Drosophila castor. And this is a transcription factor that's involved in neuroblast development during Drosophila formation. And essentially, when castor turns on in Drosophila neuroblasts, it switches their developmental potential from one neuronal cell type to another. The mammalian homolog plays a role in regulating cell fate in a number of different systems, including neuroblasts, retinal progenitors, T helper cells, and cardiomyocytes. In patients, mutations in CASI1 lead to neuroblastoma. And finally, mutations in CASI1 have been implicated in some forms of chromosome 1p36 deletion syndrome, which can in some cases include sensory neural hearing loss. So a really intriguing gene in terms of its roles in other systems. So the first thing that we wanted to do was to confirm that CASI1 is actually expressed in hair cells. So here's that same developmental trajectory. And now the redder, we go from yellow to red as CASI1 is expressed. You can see it's not blazing here, but it is expressed right about where we'd expect a phase 2 gene to be expressed. If we use SM-FISH to localize it, we can see it here in the hair cells. We use CCR2, that new marker for hair cells right here with CASI1 in green. And then at E18, we can see it's very strongly expressed in the hair cells using parvalbumin as a hair cell marker. One of the nifty things that we can do with a data set like this is we can look at expression not only of CASI1, but of some of its potential targets. So here's our P1 data set again. Here's CASI1 in both inner and outer hair cells. And these are 4 putative CASI1 targets from the literature, HCR3A, MDGA1, MYL4, and Plexin A2. And you can see they're all largely expressed in hair cells in a pattern that sort of trails CASI1 a little bit, suggesting that the CASI1 transcriptional cascade is being activated in these cells. So as a first step, what we wanted to do was to generate a CASI1 mutant. Germline deletion of CASI1 is an embryonic lethal or perinatal lethal. So we used EMX2 Cre, which is expressed throughout the cochlear duct, beginning about embryonic day 12, in combination with the CASI1 floxed allele to knock out CASI1. And this is what the mutant looks like at P1. So here's our control, inner hair cells, pillar cells, and outer hair cells. And here's the CASI1 mutant. So all the hair cells are present, so it's clearly not required for initial hair cell differentiation. We do see, however, some defects in development. So one of the things that happens as hair cells are beginning to develop embryonically is their luminal surfaces really start to expand, so they grow in size. And it looks like there's a disruption there because these cells, the luminal surface areas are smaller than in the control. And you can see that here if you quantify the data, they're about half the size that they should be. And the pillar cell space, which is another sign of differentiation, is also smaller, suggesting that there's a defect in overall hair cell development here. If we carry this through to P30 and look at the adults, we see that that defect in development seems to persist. So here's our control. Inner hair cells are here, pillar cells here, and then outer hair cells here. And now what we're seeing is rather than the hair cells being too small, they're actually too big. And that's because as the organ of Corti begins to develop postnatally, the luminal surfaces basically pinch in to flex the epithelium a little bit. And that doesn't seem to be going on here. So the hair cells are now too big rather than too small. We're also starting to see some patterning defects, as you can see here. And it looks like we're losing some hair cells. So some subtle but very clear phenotypic problems here in the CASI1 mutants. Where the story gets more interesting is when we did functional assessments of auditory function on these mice at P30. So here are ABRs, our controls are in white, and our CASI mutants are in green. And what you'll notice is between a 30 and 40 dB threshold shift at P30 across frequencies between 8 and 32 kilohertz. And if we do DPOAEs for these mice, we see essentially no autoacoustic emission. So here's our control, and these are our CASI1 mutants, as you can see right here, basically at the noise floor. So while the phenotype in terms of an obvious morphological deficit is not huge in these mice, there's clear functional deficit when we knock out CASI1. Now, this is about, we were moving along with this project and had gotten to about this stage maybe 6 or 8 months ago when I learned that 2 other labs were also interested in this gene, Botan Bonfils at the University of Iowa and Angelika Dulzelhofer's lab at Johns Hopkins. So the 3 of us got together and we decided that we would collaborate on this with Botan taking the lead. And so Botan's lab has been moving forward, looking at these mice in more detail. As far as we can tell, they start to develop stereocellular bundle defects fairly soon after P30, and they start to have hair cell degeneration fairly soon after that. And Botan is in the process now of doing RNAseq to try and understand what genes are being regulated by CASI1 and the roles of those genes in maintaining hair cell function and survival. So we're excited to find out where this story goes as we move forward. But a nice example of how we've used bioinformatics to identify a candidate gene that turns out to be important for hair cell formation and may have an impact on patient populations as well. Okay, I'm going to finish up with one last story about another zinc finger, and that is, I don't think this one turns red. That is this gene here called MYT1 or MIghty1, stands for myelin transcription factor 1. Again, came up here in our bioinformatic analysis and also in our ATO1 subtraction. If we look at MYT1 expression at either E16 or P1, here are the hair cells at both those time points. And you can see expression of MIghty1. It's fairly weak, but it's very specific here to the hair cells at both time points. We were interested in this gene because, again, because of its known function in other systems. In this case, during Xenopus development, MIghty1 has been shown to be important for allowing cells to develop as neurons in the neural plate. And it seems to do that by providing an autocrine-based inhibition of notch signaling. So basically, it allows cells to overcome notch inhibition and differentiate as a primary sulfate. That was interesting to us for inner ear because in the inner ear, hair cell is the primary sulfate, the one that prosensory cells seek to overcome notch signaling to develop. So we were interested to see whether MIghty1 might be playing a similar role in notch modulation in the inner ear. As before, we confirmed that it was in fact expressed in inner hair cells using SM-FISH. This is E15. In the apex, there's no expression. We start to see expression here in the base ahead of, this is, so parvalbumin, so this is the first inner hair cell. You see somewhat broader expression of MIghty1. It then resolves to the hair cells at embryonic day 16, both base and apex. And by postnatal day 1, you can see that it's starting to go away. So it looks like it's actually going down here. So a very nice recapitulation of what our single cell data showed us in terms of a transient expression of MIghty1 during development. MIghty1 is also a perinatal lethal. So we again use EMX2 Cree combined with FLOX to generate a mutant. At a gross level, those cochleas look pretty normal. So here's our control, EMX2 plus with MIghty1 FLOX. And then here's the mutant using EMX2 Cree to drive deletion of MIghty1. The overall cochlea looks pretty normal. But if we zoom in and look at the epithelium, we see a phenotype that's consistent with mild loss of notch signaling. So again, here's our control, inner hair cells and outer hair cells. And in the MIghty1 mutant, we see overexpression of both inner hair cells that you can see here and also outer hair cells, which you'll see here, basically 4 rows of outer hair cells here. So as I said, this is a phenotype that's consistent with a mild loss of notch phenotype. If there were no notch signaling, basically every cell would be a hair cell. There'd be no supporting cells, and we're not seeing that. But we do see an increase in the number of cells that are forming as hair cells. If we quantify this data, we can see that there's a significant increase in the number of extra hair cells, and as is typical for the organ of Corti, that's significant only in the mid and apical regions. We're actually getting pretty normal formation in the base. As I said, quite often when you see a notch phenotype, what will happen is you see a gain of hair cells at the expense of outer hair cells. So we wanted to look at the outer hair cell population. So we used a different marker, S100A1, which labels the outer pillar cells and the Diter cells here in the control. Oops, and that's one ahead. And here's the MIghty1 mutant. So we certainly have plenty of supporting cells that are here, as you can see. But if we look at this region that's mispatterned and zoom in, we see examples of regions where there appear to be two hair cells touching. So right here and right here. So there does appear to be a defect in supporting cell formation. And we're going back in now and looking to see whether there may in fact be more supporting cells missing. And that some of these lumen projections that we see are supporting cells that are trying to do double duty and essentially prevent more than one pair of hair cells from touching one another. Also, we're currently aging these mice out to see whether there's effect on, see whether deletion of MIDI1 has an effect on auditory function in any way, shape, or form. Okay, so to finish up, as I told you, my lab is interested in trying to understand how prosensory cells make these different choices. We've been using single-cell RNA sequencing to try and build out these different transcriptional programs. So far, we've really only made good progress here on outer hair cells, although most of what I showed you probably applies to inner hair cells as well. We've been able to identify these 4 phases of outer hair cell formation. And I've shown you data on 2 new genes that we've identified that are important for outer hair cell formation, CASD1, which plays a role in outer hair cell differentiation and function, and MIDI1, which seems to play a role in sorting hair cells versus supporting cells. Obviously, there's a lot more work to do. As I said, our list is not comprehensive. You're going to hear, I think, a number of other talks about this particular developmental pathway today, things that we're not working on, but other labs are doing a great job with. And there are all these other cell types that we still need to figure out what's going on with. So a lot of work still to do, but single cell omics has really given us the tools now, I think, to start looking at these other cell types. And finally, I want to thank the folks who did all the work that I talked about, or almost all, and some of the bioinformatics myself. Betsy Driver is the lab staff scientist. She handled the ATO1 mutant RNA-seq experiment, and then headed both the CASI1 and the MIDI1 projects with help from 2 former postbacks, Rebecca Hipp and Abigail Donaldson. Braulio Peguero, a current member of the lab, did all the auditory functional assessments that I talked to you about. Mike Kelley and Lakitha Kolo did the single-cell RNA sequencing from cochlear development that I mentioned to you, with help from Alejandro Anaya Rocha, who's a really good histologist and did a lot of the SM fish. I want to mention the NIDCD sequencing core headed by Rob Morell, which is incredibly helpful for all of our sequencing that we do. Hui Chang is a bioinformaticist at NIDCD that helped with some of the scenic and David analysis that I did. And then Rona Hertzano, a longtime collaborator of mine, whose lab is now around the corner from mine, who created the GEAR platform, which has been incredibly helpful. And about half the images I showed you were done on GEAR by me, because GEAR is so easy to use. And then just a few current collaborators, most relevantly Joe Burns, who actually has left Decibel Therapeutics to start his own business, and then Botan Bonfi at the University of Iowa, with whom we're doing the CASI1 project. And with that, I will thank you for your attention, and I'm happy to answer any questions.
The left side of the podium, any questions? Oh, so there's a microphone. Sorry.
CASI1, was it outer hair cell specific? I thought it was a general hair cell.
It's a general hair cell one. We didn't have enough inner hair cells to do a great bioinformatic analysis, so we did outer hair cells, but not surprisingly, a lot of the genes that we found ended up being for both hair cell types.
And maybe in the 3rd phase, you think maybe you'll have.
Yeah, we hope those will be more outer hair cell, but INSM1 comes up in phase 2, so there are clearly some in both.
Maybe this is some of off topic, but I was surprised to see that you showed MPY in the cochlea. It's a brain peptide regulates food intake. So any comments on its role?
No, just the same thing. We were baffled. We're like, what is this thing doing here? Why are they expressing this? There's an NPY mutant mouse that I think has basically no phenotype, so that's not been particularly informative. So we don't know what it might be doing there. Those same cells express NGFR1 as well, the nerve growth factor receptor. So there appears to be some sort of neuronal type of phenotype in those cells, but as far as we know, they're not innervated in any way, and so we don't really understand what is going on there.
Just from a very practical point of view, one of the limitations I find in the field is not having good antibody markers to the different supporting cell types. It would be great. Based on your RNA-seq, you probably are already testing some, right? So something that distinguish titer cells versus others, or titers 1 and 2 versus 3, or, you know, we have a marker for inner pillars and a marker for outer pillars, and it's not very good.
Yeah, it's been very useful for that. And you can, right, all the data is on gear, so you can actually go and look yourself if you want to try and find some new markers. Just so that not for titer cells, not for discriminating titer cells rows 1 and 2 from 3. But I would expect there are some there. We just haven't looked that closely at that. The NPY and the ACE1, we have antibodies for both of those that work really well. And there's a great NPY Cree line that is really good for inner pillar cells, for knocking things out.
Any other questions from the audience? If there's no more, let's thank Dr. Kelley again for his very insightful talk. Thank you very much.
Thank you.